Have you ever really thought about how your body, well, builds itself? Today, we're going to shrink down, way down, and take a look at the incredible microscopic factories that are running inside every single one of your cells, building the very things that make you, you.
Right, so you've got trillions of these little factories, and each one has a blueprint, a really detailed set of instructions. So the real question is, how does the machinery actually read those instructions and turn them into something as complex as a protein? You know, the first hurdle the cell has to clear is basically a language barrier. The genetic blueprint is written in one chemical language, but the final protein is built using a totally different one.
So on one side, you have the blueprint. It's a molecule called messenger RNA, or just mRNA, and its language has only 4 chemical letters. But on the other side, you've got the finished protein, which is built from a set of 20 different building blocks, amino acids. So it's kind of like trying to translate a 4-letter alphabet into a 20-letter one. It's not a simple one-to-one swap. The cell has to be really clever about how it reads the code. The thing is, the cell reads this code without any commas or spaces. It just goes. So imagine the genetic sentence is, "The cat ate the rat." Now look what happens if we just jam one extra letter in there, that G. The whole reading frame gets shifted, and the sentence turns into complete gibberish. That's exactly what happens inside the cell. One tiny slip-up can make the entire protein totally useless.
So how does the cell get around this? Well, it reads that 4-letter alphabet in these non-overlapping 3-letter words, and we call these words codons. The factory itself, this amazing machine called the ribosome, just chugs along the mRNA blueprint, reading it 3 letters at a time. Each 3-letter codon, like AUG or CAG, is a very specific instruction for one particular amino acid.
Okay, so the factory can read the instructions. Great, but now for the next massive challenge. The factory floor is this chaotic soup with millions of different parts floating around. So how does the ribosome grab the exact amino acid it needs at the exact right time?
The cell has this incredibly sophisticated delivery system. Think of it this way. The ribosome is the factory, and the mRNA blueprint is running right through it. Specialized molecules act like little delivery trucks, bringing in the parts. And one of these trucks, a protein called EFTU, grabs a specific amino acid and drives it over to the factory's loading bay, a spot we call the A site. And here's the key. It only unloads its cargo if the part it's carrying is a perfect match for the next 3-letter word on the blueprint.
And this is what's just mind-blowing. The system is unbelievably accurate. The ribosome gets it right more than 99.99% of the time. The error rate is something like 1 mistake for every 10,000 parts it adds. I mean, how is that even possible? Well, the secret is a really clever proofreading step. Before the factory locks that new part in for good, there's a tiny little pause. It's like putting a key in a lock. If it's the perfect match, the key fits snugly and stays put, but an incorrect match? It's a wobbly, unstable fit. It doesn't connect right, and it just kind of drifts away before the factory has a chance to permanently attach it. Pretty smart, huh?
All right, so we've got the correct amino acid delivered and held in place. Now the factory faces its most important job, forging a permanent, super strong chemical bond to link this new part to the growing protein chain. This whole high-energy reaction is carried out by the factory's central engine, an enzyme called peptidyl transferase. This is the catalytic heart of the ribosome. It's the part that does the real work, snapping these amino acids together, one after another, building that protein. Now, for decades, scientists just assumed this enzyme had to be a protein. I mean, almost all enzymes are. But when they finally got a really high-resolution look deep inside the ribosome's core, they found something that turned our whole understanding of life on its head. This quote from one of the big studies really says it all.
Yeah, let that sink in for a second. The core machine that builds all the proteins isn't a protein itself. It's a piece of RNA. This was a revolutionary discovery. It proved that RNA isn't just a simple messenger molecule. It can be a machine, too. This type of catalytic RNA, called a ribozyme, is like a fossil from the dawn of life, hinting that RNA might have been the original do-it-all molecule, storing information and doing the heavy lifting. Let's move on to the final problem in this whole assembly line cycle. So a new part has been added. Now the entire assembly line has to shift forward exactly 3 letters to read the next instruction on the blueprint. How does it do that? This big mechanical step is called translocation. Another protein, EFG, comes onto the scene, and you can think of it like a powerful lever. It uses an energy molecule to bind to the ribosome, and it physically shoves the whole system, the blueprint and the growing protein, over by exactly 1 codon,
just 3 letters. And that clears out the loading bay, the A site, getting it ready for the next delivery truck to pull in.
But how does this lever, EFG, know exactly where to grab on to get the leverage it needs? Well, the answer is this amazing trick that evolution came up with called molecular mimicry. It turns out the 3D shape of the EFG lever is almost identical to the shape of the EFTU delivery truck we talked about earlier. It's kind of like having 2 completely different keys that somehow fit the very same lock. This mimicry lets EFG dock right into that empty A site, and it uses that position as the perfect leverage point to shove the whole assembly line one step forward.
So that brings us to the complete process, this elegant, powerful 3-step cycle. First up, delivery. The EFTU truck brings in the right part. Second, bonding. The ribosome's RNA core forges that link. And third, translocation, where the EFG lever shifts the line forward. And this cycle just repeats over and over and over, adding amino acids at a crazy speed. We're talking up to 20 every single second. This whole microscopic assembly line is just a masterpiece of precision, building almost perfect proteins trillions of times a day inside you. But that near perfection also happens to highlight its greatest weakness. What happens to the final product, to you, when just one single letter in that initial blueprint is wrong? Well, that's a question for another explainer.